Um, our next speaker is Dr. Linda Spear, and we asked her to come because when I hear students talking about stress, quite often what happens is they say, oh, I've been so stressed out, now it's Friday, I can kick back, go to a party, maybe drink some alcohol, and kind of alleviate. Yeah, sounds good. Neuroscientists do the same thing. I don't know how many times I've heard my colleagues sort of say the same thing, but we thought it would be really interesting to ask Linda to come give a talk about what the long-term effects of alcohol in the brain are, because that's something that is on our brain a lot, because we sort of think of alcohol as a way of sort of solving our stress. And then after Linda's talk, we'll have a break, and then we'll come back with some real solutions for you as far as stress goes. Linda Spear is a distinguished professor of psychology and director of the Developmental Exposure Alcohol Research Center at Binghamton University. She is also internationally renowned for the research she does, and we're very happy that she was able to join us here for our symposium. Thank you, Linda.
Great, and I've got the slides on and everything. That's super. So I am going to talk about adolescent alcohol, and I'm going to build in a little bit about stress at the end, because it turns out that use of alcohol may increase stress responsivity and sensitivity to stressors, and stressors themselves may increase alcohol use. So it may be a positive feedback system that I'll talk about a little bit at the end. To get started, I use an animal model of, am I pointing to the wrong thing? Okay. I'm using an animal model of adolescence in the rat. And the reason why I think that it's possible to use an animal model of adolescence is because if you consider adolescence to be the transition from dependence on parents or whatever to relative independence, that's a developmental transition that all mammalian species go through. Interestingly enough, there's common goals that are associated with this. The individual has to learn to live independently, essentially, to find a mate and mate and have offspring to pass on the genes to the next generation. So it's a variety of common goals that are associated with that. During adolescence, there are very similar biological changes across a variety of species. Puberty occurs at some point in time during adolescence. Adolescence is usually pretty broad, and puberty's a period of time within that broader interval. There's also a characteristic growth spurt that occurs during adolescence of all species. And amazingly similar brain changes occurring throughout the brain. And the kinds of changes that we see that are very prominent during adolescence are changes in prefrontal cortex, changes in the amygdala, changes in the nucleus accumbens, changes in the hippocampus, other limbic regions, very similar to the regions that Steve was talking about. Those are undergoing remodeling during adolescence in human adolescence and in other species of adolescence as well, like rats. And associated with those brain changes is a variety of age-specific behavioral features that are also seen in adolescence of a variety of species. For instance, there's increases in peer-directed social behaviors, and increases in risk-taking. There's even increases in fighting with parents that you see not only in human adolescence but in some other species as well. And this may extend not only toward natural drugs, toward natural experiences, but also be associated with drug and alcohol use. You can see elevations there as well, as we'll be talking about. One of the problems with adolescence, though, is adolescence, it's not like a day comes and you're in adolescence, and then there's another day and you can say, well, that person's out of adolescence. It kind of something you slide into and kind of slide out of. And so it's really difficult to precisely define the duration of adolescence. In humans, it's sometimes considered early mid. Adolescence would be considered 10 to 18 years, or sometimes people say 12 to 18 or something like that. And late adolescence, emerging adulthood would be 18 to 25 years, with females tending to move through adolescence earlier than males, and males tending to take longer to get out of it than females. It's the same thing in rodents, only at the time it's really ratcheted down. So we're talking about a 2-week interval from 28 to 42 days for early mid-adolescence, and then from 42 to 55 or so late adolescence period. And in primates, it really varies with the species. Sometimes they call it older juveniles or subadults, and they don't even really use the term adolescence. But it's important to recognize that although we can model certain aspects of adolescence in rats, that doesn't mean we can model anything. It doesn't mean we can model everything in adolescence that we see in humans, that we can model them in rats during adolescence, or really during any point of time, actually. But we think that this model may be useful. So as you go through listening today, is what we're talking about in terms of what we're seeing in our animal model of adolescence in the rat. You need to think about how applicable this is to humans, and I'll be showing you some data from the humans towards the end. Okay. This is an example just showing you that during the adolescent period, you see these characteristic increases in sensation seeking here, novelty seeking in rats, that are occurring during early mid-adolescence. And these are just some examples. There's lots and lots of data like this. The data on the left are from Laurie Steinberg's lab, and the right is from our lab. So they're very characteristic responses that you see peaking in adolescence across a variety of species. Interestingly enough, one of those is alcohol use. And to say that the increased incidence of alcohol drinking and binge drinking during adolescence occurs in other species implies that there must be a biological component to that. I mean, obviously, in humans, there's a lot of reasons why kids drink and college students drink. But part of it may be biological. And because not only human adolescents, but also their animal counterparts, all in the red, show increased incidence of alcohol drinking. This is per occasion drinking on the humans, and episode drinking also in the adolescent rats on the right. And so that's elevated over adults. So you see this greater intake of alcohol in adolescents. Now, one of the problems is adolescents eat and drink a lot. And so it could be not just alcohol, it could be just anything. So we did a study where this is preference data. So anything with a 0 or above means they drank more of the sweetened alcohol solution. Adolescent rats, just like humans, don't often drink straight vodka. You know, they're into the mixed drinks. And beer, they like beer too, as do human animals. And so you see the adolescent rats are showing a considerable preference for the sweetened alcohol, whereas the adults are not. But it could be the sweetener, of course. So we also ran this just with the sweetener alone on the right. And what you can see is there's no strong preference or a preference for either the sweetener or the water solution, really at either age. So it does appear to be specific to the adolescent that they like the alcohol, and it's not just the sweetener alone. Well, why would adolescents drink more than adults? And one of the factors seems to be that they differ a lot from adults in their sensitivity to alcohol. And I want to go over these. On the one hand, adolescents are more sensitive to a few effects of alcohol. They're more sensitive to the low-dose social facilitating effects of alcohol, that social lubrication, social increase. They're more sensitive to the rewarding effects of alcohol. But unfortunately, they're also more sensitive to acute alcohol-induced disruptions in memory and in brain plasticity indexed by long-term potentiation and other measures. And that the latter finding is from Scott Swartzwelder's work at Duke. But these are the only things that adolescents seem to be unusually sensitive to. And I'm going to give you examples of the social facilitation effect. This is dose of alcohol down here. And this is a frequency of social play. And what you can see is a low-dose facilitation of that behavior in adolescents and not in adults. Not in normal adults. We never see this in normal adults. And it's important that use the word normal here because we'll be revisiting that point later. Now, the thing is, though, social play is an adolescent typical behavior. And you might say, well, maybe that's why adults aren't showing this social facilitation. But when we look at another behavior, that's social investigation, and you can see under baseline sailing conditions, this is a more adult typical behavior. Again, you see an adolescent facilitation, and you don't see that in adults. Social investigation is sniffing, basically. So adolescents are showing this social facilitation. Now, another possibility is it could be just that we're giving this alcohol, these low doses of alcohol, and it's just activating all behavior. So we did a study where we either tested them with a partner or a partner-sized cotton ball. And what we found is that they interacted with the cotton ball and drug it around and everything, but they did not show this low-dose social facilitation of that behavior like they show the low-dose facilitation of social behavior. But those are the things that animals are more sensitive to during adolescence. For most of the effects of alcohol, adolescents are less sensitive than adults. And this includes aversive effects, social inhibitory effects, sedative effects, motor impairing effects, discriminative stimulus effects, in other words, the ability to detect that the drug is in their system, analgesic effects, and hangover effects, and others probably as well. But many of these seem to have in common is these are many of the effects of alcohol that help people to determine that it's time to terminate intake, right? You start getting tired, you're slurring your words, you're motor impaired, you're staggering around. That is kind of an encouragement to stop drinking, right? So adolescents are less sensitive to those effects. And let's look at some examples of that. This is back to the social behavior, and if you were paying attention, this is exactly the same slide as I showed you before. This is the social facilitation we see in low doses in adolescents in the red that we don't see in adults. But if you look here at the higher doses induce social inhibition. And what you see here is adolescents require a higher dose to get this inhibition than do adults. Adults are more sensitive to the social inhibitory effects. You see this not only with social play, but also in social investigation. In this dose range, there's not a significant decrease in social investigation in adolescents across this dose range of alcohol. If you gave them a higher dose, they definitely would. They definitely would at some point. Whereas the adults are showing social inhibition at the two highest doses. So adults are more sensitive to social inhibitory effects than our adolescents, and these are part of the cues that are used to terminate intake. This is another study. This is a study of sedative effects. Basically, it's a study of rats passing out. We give them a dose of alcohol that's sufficiently high that they literally pass out. Then we put them on their back, and they don't like to be on their back. So as soon as they wake up enough, become unsedated enough to turn over, that's what we call this recovery time. So it's a time from the time that they can't right themselves, and we put them on their back until that they're able to right themselves. And as you can imagine, as the dose increases, it takes them longer. But if you look up here, our adults are taking twice as long to wake up than the adolescents. The adolescents are waking up much more quickly. Now, there could be a boring reason for this because it's possible that adolescents tend to metabolize things faster than adults, get them in and out of their system more quickly. Their higher metabolic rate is why they can eat more and not gain weight, whereas adults, if they looked at that much food, would gain weight. And so what we see, so it's possible that adolescents are just waking up sooner because they're getting rid of the alcohol in their system. So to look at this, we looked actually at brain levels of alcohol to determine if there was a difference. And what we see is that adolescents are not only sleeping less long, but they're waking up with higher levels of alcohol in their brains. Their brains are less sensitive to alcohol. So when they get up in these 300 and so milligrams per deciliter level, that's low enough that their brains will allow them to wake up, that they're able to wake up, whereas adults have to metabolize quite a bit more alcohol, keeping them asleep longer before they're able to wake up. It's a difference in actual brain sensitivity to alcohol. Last example I want to give you is the classical conditioning taste aversion model. And this is a really simple procedure. Basically, you expose the animal to a novel taste, and then you inject them with alcohol. If they find the dose of alcohol aversive that you injected them with, the next time you expose them to the taste, they won't drink as much of that taste because they attribute it to the negative effects. Probably you all have developed a taste aversion to something, gone out and eaten something and gotten sick, and then you don't like that substance anymore. For me, it was baklava, but everybody has their thing. And so this is conditioned taste aversion, and what we see here, this is an unusual measure, but basically any of the scores above 0. Let's focus here on the adolescents and the adults given the control solution. So they're given an opportunity to taste this novel tastant, and then they're given an injection of saline control solution. Has no effects. And what you see is the next time they're exposed to this novel taste, they drink a lot of it. At all doses of alcohol in adults, after each of these doses of alcohol in adults, they avoid that taste. So they're showing taste aversion at all these doses. If you look at the same range of doses across here in adolescents, they don't show taste aversions at any of those doses. You have to go to a higher dose range, 2- to 3-fold higher, for the adolescents to show a taste aversion that you're seeing in a much lower dose in adults. So there is all this raft of differences between adolescents and adults in their sensitivity to alcohol. In a sense, alcohol is a different drug for adolescents than for adults in terms of how they respond to it and their sensitivity to the different effects. We were interested to see if it was just alcohol or if it might generalize to other drugs as well. And if we wanted to look at that, I mean, the measures that we can look at across drugs are the rewarding effects and aversive effects, because those have been studied in a variety with a variety of other drugs in adolescents. And so we're going to focus in on those. And in terms of the rewarding effects, and that's usually indexed by a conditioned place preference, where you basically give the animals a drug in one place and give them a control solution and put them in a second place. And then you give them a number of pairings, and then you see if they prefer the place where they had the drug. To the extent that they find the drug reinforcing, that's used as the time that they spend on the drug paired place is used as an index of how reinforcing they find the drug. And what you find is that adolescents not only find alcohol more rewarding, but they find nicotine more rewarding than adults, and in cocaine and amphetamine more rewarding than adults as well. This is some of the work by our lab and other labs as well. What about the aversive effects? In this we index via conditioned place aversion, where they avoid the place that was paired with the drug, or by conditioned taste aversion, like I just showed you before. And you see adolescents being less sensitive than adults to the aversive effects of nicotine, amphetamine, and marijuana, THC, cannabinoids. And so it seems rather generalized. It's not just alcohol. This whole bias towards reward and insensitivity to aversion seems to expand and be seen across a variety of drugs, and maybe even drugs, non-drug stimuli. We did some studies where we put taste into the mouth and then looked at the mouth movements to that, and you can by the characteristics of the mouth movements, you can determine how reinforcing or aversive they find these tastes. And adolescents are more sensitive to the positive effects of positive drugs like sucrose, and they're less sensitive to aversive taste effects like quinine. So it may even extend beyond drugs. Adolescents are reward sensitive, and aversion not sensitive. The next question, though, becomes, why are there all these alcohol typical sensitivities? As I showed you before with the sedative data, it's not pharmacokinetic. It's not just how quickly the alcohol gets metabolized. But adolescents do tend to metabolize alcohol a little bit differently than adults, which means in all of these studies, looking at all these behaviors, we really have to look at alcohol levels. We also monitor alcohol levels to make sure that no one case is associated with just some change in alcohol sensitivity, and none of these instances is it related to differences in alcohol levels between the adolescents and the adults. We've also spent a long time gonadectomizing animals and looking at the role of and putting hormones like testosterone back in and looking at the impact of gonadal hormones, and that doesn't seem to be a long story short, it isn't a major factor either. We've also looked at a number of different neurotransmitters, which I'm not going to get into there, but as the data today. But there are a variety of different neurotransmitters that seem to be involved in these different effects. And these systems, again, are maturing through adolescence. And during the time that they're still continuing to mature, they may be related to these differential alcohol sensitivities. They may be responsible for these different alcohol sensitivities. But of course, this $54 million question is, how relevant are these data to human adolescents? Now, by and large, we don't have the data to be able to look at that because you can't give alcohol to young adolescents and study them empirically in experiments. So you go to the literature and see what we can glean and what we have been able to find and see how well this relates to what we've been seeing in rats. Let's first go back to the more sensitive to alcohol-induced social facilitation. Adolescents are more sensitive to that than adults. Is there any evidence from the human literature? Well, actually, if you go to survey data, there's a lot of stuff showing stuff like that. The literature suggests that most students drink primarily for social purposes. From high school survey data, the most important factors across all the measures of alcohol abuse was drinking for social facilitation. Age distinctions and effects reflect the particular importance of social interactions and drinking occasions in young people. That doesn't mean adults and older adults don't also drink for social facilitation. It's just that young people are especially sensitive to that. What about the acute disruptions in memory and brain plasticity? This is a study that was done in Scott Swartzweiler's lab. And you can't give alcohol to individuals that are under 21. But in this study, he was looking at that late adolescent, emerging adulthood. And he gave alcohol to 21 to 24-year-olds and compared the results with 25 to 30-year-olds. And in this study, he was looking at a complex figures task where basically you allow somebody to look at a figure for a brief period of time, and then later you ask them to draw that figure. And also verbal learning tasks where it's basically you have to learn lists of words. And the individuals, I almost said animals, the individuals were either given allowed to drink a placebo solution that was smelled and flavored to be like alcohol, or given a dose of alcohol that produced peak blood alcohol levels about 70 milligrams percent, which is below the binge range of 80 milligrams percent, or the DUI range of 80 milligrams percent. So it's a bit below that. And again, the data in red are going to be the 21 to 24-year-olds, and the 25 to 29-year-olds are in blue. They were given baseline tests, and then they were given alcohol, and then after the finishing learning, the acquisition trials, 20 minutes later they were given a memory task. And what you see in both instances that the 25 to 29-year-olds in the blue were not impacted by the alcohol. They were able to perform this task. But if you look at the 21 to 24-year-olds, their performance dropped off rapidly on the memory test that was given 20 minutes later. Now, these are really relatively old adolescents. One would predict that the effect would be even more striking in younger adolescents. Okay, so what about the lessened sensitivity to alcohol-induced things? I only know of one study that has addressed this. And they did a study that there's no way we could do today. Because this is back in 1983, and they actually gave alcohol challenges to 8 to 15-year-old boys. And they were producing blood alcohol levels about 34 to 35 milligrams percent. It's in the moderate consumption range. Again, 80 milligrams percent would be DUI. So it's in the moderate consumption range where you see impairment, but they're not stinking drunk. Okay. And these, are some of the quotes from that study. In the study of the first intoxicating experience, I don't know how they knew that, let's not go there. In the first intoxicating experience of naive children, little behavioral change was noted clinically, subjectively, or on a validated objective test of intoxication. These children exhibited a smaller behavioral change than expected for their blood alcohol level. None behaved grossly intoxicated as our adult subjects were. And by the way, the adult subjects were social drinkers, had quite a bit of experience over the years drinking alcohol, and presumably had already developed some tolerance to alcohol. Certainly had a lot more prior experience with alcohol than this naive group. We were impressed by how little gross behavioral change occurred in the children after a dose of alcohol, which had been intoxicating in an adult population. So this suggests that perhaps some of the things we've been seeing in our animal models of this increased sensitivity to the rewarding and the good stuff about alcohol, decreased the sensitivity to the aversive stuff about alcohol, and the intake limiting effects of alcohol that seen during adolescence might also appear in humans. But would that be a good thing? I mean, why would it matter if adolescents are relatively insensitive to alcohol intoxication? I mean, that would suggest that if they were more sensitive, that we would probably have a lot more overdose deaths than if they were less sensitive. So why isn't it a good thing? Well, the concern apparently seems to be that being insensitive to the intoxicating effects permits and encourages heavier drinking. Indeed, a low initial sensitivity to alcohol is a known risk factor for problematic alcohol involvement. And that is associated with an increased risk for later alcoholism. And a low initial sensitivity to alcohol is seen in a number of instances. It's seen genetically. It's very well established by Mark Shuckett and others that individuals with a family history of alcoholism, most typically they study male kids, offspring of male alcoholics, show insensitivity to alcohol intoxication. You also see that in strains that are bred of mice or rats that are bred for high alcohol consumption and preference. They also are very insensitive to the aversive and other effects, intoxicating effects of alcohol. If you believe that rat data, it suggests that this insensitivity is normally seen to some extent during adolescence. And there's some evidence that prior experiences, particularly stressors, may decrease sensitivity even further. And I'll show you some of that data in just a little bit. So this may result in these kind of double and triple whammies that really permit really high levels of use. If you've got somebody from a family history of alcoholism who has a stressful life existence, they enter adolescence, start drinking, become even more tolerant, you're really driving these high intake levels, and that may cause problems. Part two to why I think that adolescent alcohol sensitivities may contribute to problematic use is related to the other side, the more sensitivity that they're showing. This increased sensitivity to rewarding and social facilitating effects of alcohol can help promote drinking. They like it, it's rewarding, it facilitates their social behavior. And indeed, an increased sensitivity to ethanol reward and stimulatory effects is also seen genetically in family history positive offspring, and developmentally, if you believe what we've been talking about in the rats, normally during adolescence, and as a function of prior experiences and stressors. So they show more of the positive stuff and less of the negative stuff may help enhance intake. Let me show you some of the stress data. This is a play-fighting behavior in animals that either were not stressed or were given 5 days of restraint stress for a short period of time each day. And what you see in the control animals is the usual alcohol-induced social facilitation and social inhibition at higher dose. In the stressed animals that were stressed every day and then given the alcohol challenge, what you see is they're showing even more social facilitation for over broader dose range, and they're less sensitive to social inhibiting effects. So they're becoming even more, that tip of that balance is tipping even more towards positive and away from aversive effects, which could enhance alcohol intake. So stressors appear to increase these natural developmental biases towards increased appetitive and decreased aversive effects. So these patterns of increased and decreased sensitivities could serve as permissive factors to promote drinking, and these patterns might be even more exacerbated in people that are vulnerable, either because of the genetic history or because of the prior experiences like stressors. And indeed, one of the things I've been shocked about is really how much the adolescents are drinking. And there's very little field data on this, but we, in conjunction with several clinical psychologists at Binghamton, we have been running field work and looking at human adolescents. The KEG means Collaborative Ethanol Group, cute acronym, right? And what this study involved is we went to downtown Binghamton on Thursday and Friday nights. Saturday night's date night. So we went on Thursday and Friday nights because they were typically out drinking with friends. There's something like 8 bars in a 1-block area. And we had pairs of research assistants that were very outgoing and friendly go up to people as they're bar hopping, going from bar to bar, and ask them, would you be willing to fill out a short survey? And, well, it was actually probably 8, 10, 12-page survey, but many of them were. I mean, because these are, like you can imagine this like group of 4 guys, and these 2 girls, outgoing girls, come up and say, "Would you fill out the survey?" And the guys would get, "Yeah." There was no reinforcer or anything, but we provided them the opportunity, other than the opportunity to talk to these people. So they filled out the survey, and then a randomly selected number of them, based on if their survey had a little notation on it, were invited to go into the tent. And this is the tent. This is tent testing. And in here, we did motor impairment, cognitive impairment. We got breathalyzed them at the time they entered the tent and when they left. And we also got DNA samples. So we collected these data for, I think, 3.5 years. Of course, we're in Binghamton, which is probably like Burlington, so this is an outside situation. So we're not testing the tent in December and January and February, right? So we would test in the early fall, and then once it warmed up in the spring. I'm going to show you some average data from one of these studies. You can see that the age was about 21. About half the individuals were under 21. About half of them were over. Most of our sample was about 23. When we kind of dropped people that were 30 or 35 because they were a little unusual to be in this downtown college bar area. You can see that the average number of drinks, and these are not exit interviews. These are people that are bar hopping. Some of them were pre-game, and we would count that, and they come down and go from bar to bar. It was almost 8, and the average blood alcohol levels were over 90 milligrams %, which is past DUI. I was really shocked at this. If you look at the 80 milligram per kilogram cutoff right here, you can see that literally half of the individuals were in that drinking range. We also collected, amongst the things that we checked in the survey, and we did motivations for drinking, when did you start drinking age-wise, and all that kind of stuff. Got a lot of information about it. We also administered the audit, and that's the Alcohol Use Disorders Identification Test. That's what audit stands for. And the score is from 0 to 40. And it has 3 subcomponents that are related to intake, dependence, and problems. And any score above an 8 is thought to reflect hazard or harmful use. And if you look here at the percentage of the number of individuals, the percentage of individuals that had scores above 8, 3/4 of the sample population that we were dealing with had scores in the harmful, hazardous use level. So over half of them are getting into the DUI range, and they're coming in with these really hazardous use levels. I should note here, and if you might think, well, they're drunk. How reliable are their audit data? We do have retest ability data on the audit, and it came in at 0.8 something. I think it was 0.87, which is really highly correlated with their follow-up data as sober follow-up data. The other thing that I found was really interesting is that the audit scores that we got were correlated with the age that they started drinking, not their level of intoxication at that time. So it was so the longer they drank, the more likely that they were, and the earlier that they started drinking, the more likely they were to come in with these higher audit scores, which suggests that there may be lasting effects of that alcohol exposure. And we have been looking into that, and I'm going to go over that data in a bit. So indeed, there's some potential adverse effects of adolescent alcohol use during adolescence. It may have, it can certainly impact the ability to function during adolescence. First of all, there's that greater sensitivity to alcohol-induced disruptions in memory and brain plasticity. The disruptions in brain plasticity we've seen from animal studies, and we'll talk about later, they're lasting. So this could affect ability to perform. There's also this mutual synergism. When you drink alcohol, increases risk-taking. The increased risk-taking could involve alcohol, so you do stupid things under alcohol. You drink with somebody, you drive with somebody who's been drinking a lot. You have unprotected and unattended sex and things like that. So that's a possible adverse effect during the exposure period. There's also a number of clinical psychologists have argued that adolescent alcohol drinking can get to the point where it works to kind of circumvent learning normal developmental tasks. Like if you may never learn to speak to girls unless you've been drinking, and that's kind of helping you to be able to learn to speak to girls, and so that you may be not learning coping things that you should be able to do otherwise. And also, there is some evidence in our simple animal models that this exposure to alcohol during adolescence, at the time you're being exposed to alcohol, it may alter your ability to adapt to stressors. And I'm going to show you some data here. Okay, so this is animals that are allowed to drink alcohol in their home cage. They drink a lot. Adolescents drink more than do adults. And during that period of time, they're after several days of alcohol exposure, they're given access to a stressor. In this particular situation, is foot shock. And we're looking at the amount of foot shock over these first 5-minute time bins. So it's a 15-minute session. This is the first 5 minutes, second 5 minutes, and third 5 minutes. What we're looking at is the time spent freezing. And what you'll see in adults is they increase their time freezing from the first to the second 5 minutes because they first freak out and they try to get out, and they learn that they can't escape. And so they increase their immobility. This is a very characteristic response in normal adults, and you see it in adults that are drinking alcohol in their home cage as well. You also see that in adolescents as well, that are drinking just the sweetened solution alone in their home cage. But in animals that are drinking alcohol in their home cage, they don't show that increase. They don't show that stress adaptation, the increase in immobility. So it's as if the stress is decreasing their capacity to adapt to the stressor. So this may be some sort of feed-forward system, and I tried to write, this is a really bad picture, I'm not very good at this, but if you have an individual that's stressed, and that stress, as we just talked about earlier, increases adolescent typical ethanol sensitivities, makes them more sensitive to the good stuff, even less sensitive to the bad stuff. That may serve to increase their intake. That increased intake may decrease stress adaptation, which increases their sensitivity to stress, which increases, so it keeps going around here. It's a feed-forward positive system. And that has been little studied in the human literature, the evolving human literature in this area, but it's a concern that's been raised by the animal literature. Now, what about, those are all sort of short-term consequences during adolescence, during the alcohol exposure. What about later consequences? And these can best be studied in animal studies because we can give the drug and we can wait until well into adulthood, long time after we've terminated alcohol exposure, we can see if there's any lasting consequences of this. And there's a national consortium that's called the NADIA, or the Neurobiology of Adolescent Drinking in Adulthood, that's been funded by the NIAAA to look at this. And they found a number of reliable, long-lasting consequences of this adolescent exposure. There's a number of neural alterations, including decreases in neurogenesis, the formation of new neurons. So after adolescent alcohol exposure, long time later, in adulthood, well into adulthood, their brains are generating fewer new neurons. And there's also decreases in certain brain structures, like decreases in cholinergic forebrain neurons and forebrain, basal forebrain. So there's all these neural alterations that are showing up. Behavioral affective effects that are seen, there's increases in risky choice, a lot of disinhibition that's been reported by the NADIA group. Increases in anxiety measured a number of different ways, including social anxiety, and we've done a lot of that behavior ourselves. The NADIA has also found, across the NADIA consortium, they've looked at a lot of different measures of learning, and by and large, long-term impacts, there's not long-term impacts on learning. But there are specific alterations that are related to prefrontal cortex deficits. There's decreases in cognitive flexibility, the ability to reverse on a reversal task, or to set shift from one type of paying attention to one type of stimuli to another, or the ability to extinguish behaviors, all appear to be disrupted in these individuals. And what I find particularly intriguing is after adolescent alcohol exposure, adults often, not always, definitely not always, but often retain adolescent-type characteristics into adulthood. So they act like adolescents in adulthood after alcohol exposure during adolescence. This is seen anatomically in terms of spine formation in particular brain regions. It's seen electrophysiologically, if you actually measure like long-term potentiation, that brain plasticity. They show that as well. It's seen in terms of behavior and in terms of drug response. So you see it both at baseline and in terms of response to alcohol. I wanted to, for my example of that, I wanted to go back to the slide, right? This was the one we've seen. Adolescents are more sensitive to this and less sensitive to these effects of alcohol. Which of those are also seen in adulthood when they're not normally seen in after adolescent alcohol exposure? The circled ones are seen in adults. Now, the minuses are cases, negative cases. So it's not always seen. Not everybody has reported this all the time. But in many of these cases, and I was really actually stunned about this, Scott Swartzweiler and I were surprised enough about these data that we actually wrote a review talking about this, because this seems to be a fairly common theme of the adolescent chronic exposure lasting effects, that in adulthood they're acting like, they have an increased propensity to act like adolescents. So the bottom line is that basic science studies in rats have shown that adolescents are less sensitive to many of the cues that might serve to moderate drinking, tell you to stop drinking, and more sensitive to cues that are the good stuff, social facilitatory and rewarding effects. And that these kinds of alcohol sensitivities may help promote high levels of intake, and that high levels of intake is seen in the field, and that may lead to lasting adverse consequences. And that evidence is building in basic science studies of these lasting consequences, and those include the perseveration of adolescent typical alcohol sensitivities into adulthood. I do have to thank my people, because without these people, none of this would have happened. The individuals in green are the people that did the human studies, and the others are the people in my lab, and I'm very grateful to the support of NIAAA, who's put up with me for many years. Thank you very much.